Hello folks, welcome to this third video about inheritance problems, and we're now really getting into some of the more complicated questions you might face, because having looked at monohybrid crosses, test crosses and codominance, we're now going to consider something called a dihybrid cross. And as the name would suggest, di means two. These problems are where you look at two traits simultaneously. Now, you'll often see them phrased like this. So this is the classic example. This is Mendel's experiment that he did in the 1800s with peas. So you can actually look at the seed colour, and there's two alleles, dominant, recessive, the usual relationship. But you've also got seed shape determined by a completely different gene with two alleles itself. So these are two traits that we're going to look at simultaneously. And we're going to imagine that these two genes are on separate chromosomes to start with. And the terminology for that is you say they are not linked. The problems are exactly the same. You're going to be working out the phenotypes of the offspring and the ratios in which they would occur. But in this first example, what we're going to do is we're going to cross a heterozygote with another heterozygote. Now, of course, when we use the term heterozygote, we could be referring to either of these traits. I actually mean for both traits. So in this cross, both of these individuals are going to be heterozygous. So that means that they're going to be heterozygous for trait Y, the seed colour, but they're also going to be heterozygous for trait R, the wrinkly or round. Okay, so those are their genotypes right there. Now, in order to understand how we do this, how we actually unpick what's going on here, we're going to go to one side for a second. We're going to start thinking about meiosis again. Now remember, we did this at the very start, and we said, right, when a cell's getting ready to undergo meiosis, the chromosomes are going to line up in the middle in metaphase. I'm going to use different colours for the chromosomes that we've got. I'm going to do one big chromosome and one little chromosome, and I've used orange and green. Now I've used that to show these are homologous pairs. That's one homologous pair, that's another homologous pair. And the orange chromosomes, let's imagine that those are paternal. That means that this individual, the individual who owns this cell, got both of those chromosomes from his father. And these could be maternal, and therefore he inherited both of these from his mother. Okay. Now, this is metaphase 1 of meiosis. They've lined up in homologous pairs down the middle, and we know what's going to happen. The cell's going to divide by pulling them. Here you go, there's some spindle fibres. Pulling them apart, we're going to end up with this kind of setup here. See that? And the same thing happening there. And then they're ultimately going to divide, and you'll end up with two cells produced. This is our first division, and those cells will be truly diploid. Okay. Now let's think about what this means in terms of the genotypes, because let's put some genes on these chromosomes. Now remember, we've got two genes. We've got the R and the Y. Let's imagine that the Y gene is found right there, and we had heterozygotes, so that's one capital and one lowercase. And let's imagine that the R gene is found on the smaller chromosome there, like that. Okay. Now look at what's happened as we pull these apart. My two orange chromosomes go this way. They actually, by coincidence, they both have the same dominant. They both have the dominant allele for their gene on them. So that means we're going to end up with a capital Y there, and capital R there. You might say that the genotype of this gamete is YR, and this one's got the lowercase y and the lowercase r, so we've ended up with the lowercase y and a lowercase r there. So the genotype of that gamete is lowercase y, lowercase r. That's from a parental cell that had this genotype, heterozygous for both traits. Remember those? Okay. And we've ended up with haploid gametes. Now, the classic error that people do when they are doing meiosis problems like this, or when they're doing dihybrids, is they get awfully confused about what's going on. They've got their cell, they know that it's this, and we end up with gametes with frankly bonkers genotypes like this. That's the classic error. People get awfully confused about how they're dividing their cells. Now, if you think about what that would actually mean, that would mean that we've taken our chromosomes like this, and instead of putting a big and a little one into each of the daughter cells, we've actually divided it this way, and you will have ended up with two chromosomes. It's the little chromosome with R's on it, isn't it? Two chromosomes there with the R's on them, and then the two chromosomes with the Ys on in there. That's not right. That's not how meiosis works. These are not viable gametes. So just watch out for that. That is the most common error people make. So you should be ending up with haploid gametes. By haploid, they've only got one of each letter, but they do have one of each letter. Now let's just quickly have a little look at this, because we made these gametes here, but that's actually not the only way we could have done this. If I put that to one side, and let's just reset things on this whiteboard here. Because that lining up in the middle is random, we call that random assortment, independent segregation. I put both orange ones on the same side the first time I did it, but I could have done it this way round. Now remember which genes we had. So we had a capital Y on that, and we had the capital R on that, lowercase y, lowercase r. Now if you look at what happens when we pull these chromosomes apart, you're actually going to make different combinations. Look at the genotypes of the gametes we formed here. So when it comes to dihybrid problems, if you've got heterozygous, you've actually got 1, 2, 3, 4 possible gametes when the parent is heterozygous for both traits. It's worth bearing that in mind. Obviously, if the parent were homozygous for both traits, then you wouldn't have as many gametes possible. But it is perfectly possible to get up to 4 different gametes. So my advice for you when tackling these problems is to really think about the gametes. Really, really concentrate on what can be made. You don't have to draw it out like I just did. You could, let's give you a slightly different problem. You could just look at the genotype of the parent and think, right, I need to get a Y and an R. What can I produce in terms of gametes? So both gametes are going to have capital Ys there. They could have a capital R or a lowercase r. This one, because it's homozygous for one trait, can only make 2 gametes. Let's take a completely made up one. Let's go. Okay. You don't have to draw the diagram, but you do need to think, right, I need an A and a B in each cell. Okay, so I need A, I could do capital A, capital B, I could do lowercase a, lowercase b. That's not the only combination. I could do capital A, lowercase b, I could do lowercase a, capital B. There are 4 possible combinations there. The quicker you can do this kind of visually by eye, just looking at them and figuring out which gametes you can make, the quicker you'll be able to solve these kinds of problems. Back to the example in hand. Now I told you that we were going to cross 2 heterozygotes. So what you've got to do is you've got to figure out what gametes you can make. So let's look at these gametes first of all. So they are going to be capital Y, capital R, capital Y, lowercase r, little y, capital R, little y, lowercase r. Okay, those are our gametes right there. We're going to do exactly the same for the other parent, capital Y, capital R, capital Y, lowercase r, lowercase y, capital R, and lowercase. Right. This means that your Punnett square, it works exactly like before, but now it's a 4x4 grid. There are 16 possible genotypes for the offspring. I'm going to do the first couple for you to show you how you should be writing it out. The temptation is you go YR, YR, but that is actually harder to read quickly. The convention is that you put both Y's together, both R's together. So imagine this gamete fusing with that one. That's the genotype of the offspring. Haploid gamete plus haploid gamete gives you diploid offspring. Let's pick another one at random. Let's fuse this one here with this one here. So we've got lowercase y there, we've got lowercase y there. And we've got capital R there, capital R there. So that's the genotype right there. Now I'm actually going to pause the video because you don't want to see me writing out these combinations for 5 minutes. It takes a bit of time. You've got to be really careful to get them right. But in the second, they will all magically appear. Ta-da! There we go. Right, so it takes a bit of time, but there you go. You have it. 16 possible genotypes. Now, it doesn't mean there are 16 different phenotypes. If you think about it, we've got 2 traits, haven't we? Seed colour and seed shape. And each of those has 2 different phenotypes. So there's actually 4 different phenotypes possible here. And they are as follows. So we have got green and round. We've got green and wrinkled. We've got yellow and round. The pens are giving up on me. yellow and round, and we've got yellow and wrinkled. Now the question asks you for the phenotypes of the offspring, that's those, but they want the ratio as well in which they would occur. So in order to do this, sometimes it's worth getting a coloured pen or something that you can use to highlight maybe, so that as you go through you can sort of tick them off. Okay, it's coloured pen time. So let's do like green and round first. I'm going to choose that as this colour here. So that's the dominant. That's both dominant traits. So that one, that's got one of the dominant alleles. So that will get it. That will get it. That one, not that one because that one won't be round, but that one will. That one will. That one will. Not that one. Not that one. That one will. And that's about it. So let's count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9. And there are 9 of those. Let's choose a different colour. Let's do green and wrinkled. Okay, go for orange for that one. So for green we need a capital Y, but for wrinkled we need 2 lowercase r's. So that's one of those. That's one of those. That's one of those. There are 3. Let's do yellow and round. We'll choose this colour. Okay, so for yellow we want both lowercase y's, but for round we want a capital R. So that's one, that's one, that's one. There are 3 of those as well, and there is only one left, and it's this one right here. That's the yellow and wrinkled, and we end up with this magic ratio of 9 to 3 to 3 to 1. That is the ratio you always get when you have 2 heterozygotes for both traits doing a dihybrid cross. Pause the video and have a go at the next question in the pack. Okay, so exactly the same kind of problem. It's a dihybrid. Got 2 traits: the length of the hair, short and long. Short hair is dominant, and then we've got the colour, white, black, or grey, and that's a co-dominant one. Okay, so you've got to be a little bit careful here. So we've got one that is grey, long hair, and one that is pure breeding for black, short hair. Now, pure breeding, that means homozygous, basically. That means that the trait for black and the trait for short, they haven't got any additional alleles lurking in there. So you can assume homozygous for those 2 traits. So pause it, have a go at doing that. The answer's about to appear. Ta-da! Right, so the way to approach this is to annotate as you go through. They've given us 2 traits. You get to choose the letters. Now it's about hair length, so I've chosen H because it's obvious what's capital and what's lowercase. Long-haired is the recessive one. They tell us short hair's dominant, so long-haired's recessive, so I've used little h for that. Capital H for the short hair. The hair colour can be white, black, or grey. This is a co-dominant allele, co-dominant gene with 2 alleles, 3 different phenotypes. So I've gone with the C and then a superscript notation. So CW is white, CB is black, and if you have them together as a heterozygote, you would be grey. Okay, now that's my notation sorted. Now let's look at what we're breeding. So we've got a guinea pig with grey, long hair. So grey, CBCW, that's the only way of getting grey. And it's long hair, that's the recessive trait, so it has to have both recessive alleles, so that has to be little h, little h. And the other one is pure breeding for black, short hair. Pure breeding for black means that it's CBCB, it's homozygous for that. And it's pure breeding for short hair. So that means that even though it's the dominant trait, we know for a fact there is no recessive allele hiding there. It's capital H, capital H. Now let's work out the gametes we can produce. And what you'll notice that even though this is a dihybrid cross, it's a little bit simpler because you have CBH. That's one of the gametes we can produce. You get 1 C and 1 H. You could also have CW, but that's also got to go with a little h. There's only actually 2 possible gametes from that parent, and there's only 1 possible from this parent. Because it's homozygous for both traits, you can only make that gamete, which means that even in this dihybrid cross, we're going to end up with a nice simple Punnett square. It's a 2 by 1. So you take this gamete and fuse it with that one. It's going to be CB, CB, capital H, lowercase h, which phenotype is going to be black, long hair. No, it's not. It's going to be black, short hair. Ooh, good save. That's going to be the dominant one there is going to be expressed, so you can end up with short hair. This one, CBCW, that's going to give you the co-dominant phenotype of grey hair. But again, because you've got a capital H, it's going to be short-haired. And the ratio is going to be 50-50 or 1 to 1. And there you go. You've produced the offspring and you've worked out the ratios of their phenotypes.